Okay, oxygen dissociation curve. So this graph here on the right side is actually showing the oxygen dissociation curve. So what exactly is oxygen dissociation curve? So it is a graph. So a graph of y-axis versus x-axis. So what is the y-axis? If you read here, it's called saturation of hemoglobin in percentage. So in words, it is a percentage saturation of hemoglobin with oxygen or percentage oxyhemoglobin, okay? Versus in the x-axis, it is a partial pressure of oxygen in millimeter of mercury, okay? Or in kilopascal. So you have got two scale here. You see this this x-axis got two scale. The top is kilopascal and the bottom is millimeter mercury. Okay, so this is called the oxygen dissociation curve. So then what is saturation of hemoglobin, right? So percentage saturation of hemoglobin with oxygen, it means that for all the hemoglobin that we have in our blood, what is the maximum amount of oxygen that our hemoglobin can carry is considered as 100%. So when our hemoglobin is fully saturated, it is said that it can carry 100% of oxygen. Okay, so when it is carrying 50% of its maximum capacity, it is called 50% saturated. So our hemoglobin is 50% saturated with oxygen. Okay, so 100% means the maximum volume of oxygen that our hemoglobin can carry, all the hemoglobin in our body can carry is 100%. So if you say 70% means 70% saturated, 50% means 50% saturated. Okay, out of the total, how many percent can be transported out of total maximum? So that's the meaning of percentage saturation of hemoglobin with oxygen. So sometimes when I look at this word, I find it a bit vague and a bit difficult to remember. So I use a simple way to help me to remember. So the simple way, there are two ways, but it may not be very accurate, but it's easier to understand. Okay, so the simple way, the first one is I will take number one, percentage saturation of hemoglobin with oxygen as the percentage of oxyhemoglobin. So out of total hemoglobin that I have, how many percentage of them are carrying oxygen? So this is one of the first way that I will look at it like this. Okay, that's easy to understand. Or out of total hemoglobin I have, how many percent are carrying oxygen? Okay, but this is not so accurate, but it's easy to understand. Second way to understand is that my hemoglobin has got 4 subunits, right? 2 alpha and 2 beta globin. So altogether, one hemoglobin can carry 4 oxygen, right? So therefore, when a hemoglobin is carrying 4 oxygen, I will consider it as 100% saturated. Okay, so that means to say that one hemoglobin got 4 subunits, right? 1, 2, 3, 4, right? 3, 4. So 2 alpha and 2 beta. So when all 4 subunits are carrying oxygen, I will consider it as 100% saturated. I will consider it so easier for me to understand, right? So then when it is carrying only 3 out of 4 subunits are carrying, then I will consider it as 75% saturated with oxygen, and so on, right? So if it is like this means 50%, like this means 25%, like this means 0%, okay? But this is not so accurate because you can see that it's actually in this curve. It's not only 100, 75, and then 50, and then 25, right? It's actually a smooth curve, isn't it? So this is not very accurate, but easier to understand. The more accurate one is out of your total maximum oxygen that our all hemoglobin can carry, right? Out of total 100% is maximum. How many percent of oxygen is our hemoglobin carrying now out of maximum 100%? So that's the most accurate one. Okay, so this number 1 and number 2 is just to help you to actually understand better. But the best one is still the definition. Okay, the definition. Percentage of oxygen out of the maximum oxygen that hemoglobin in the blood is combined with. This is more accurate. So I don't want to go to detail because there are more important things I need to elaborate. So we'll leave it as it is. Okay, we'll leave it as it is. Okay. Okay, so what is partial pressure? Now, concentration of gas, right? Concentration of gas is actually more accurately represented by the term partial pressure, because gas, concentration of gas is actually in the form of pressure. The more pressure you have, the more molecule of the gas that you have. Okay, or the more molecule of gas you have, the higher is the pressure. Okay, so therefore we use partial pressure to represent the concentration of gas. We use concentration only for solution. So for gas, from now onwards, you use the term partial pressure. Okay, so partial pressure is a measure of the concentration of the gas. Now you look at this oxygen dissociation curve. The curve in the graph is an S-shaped curve. So how do you describe this S-shaped curve? Either you write it as S-shaped curve or sigmoidal curve. So when you learn biology, you need to pay attention to the detail of the word. Is there a capital letter? Is there a dashed line there? You have to write it accurately. Is it a continuous word joined together or not continuous? For example, semi-conservative. Do you write semi-conservative as 2 words or 1 word, or 2 words and then linked together with a dash? All these are detail you need to learn, okay, accurately. Pay attention to this when you study. Okay, so now let's go to the detail. This graph is actually showing, it is showing this equation here, which is actually the loading and unloading of oxygen, depending on the concentration of oxygen in the surrounding. Okay, so I've already mentioned that when oxygen level is high, okay, high oxygen concentration, I'm using concentration, I'm going to use, okay, partial pressure of oxygen, partial pressure of oxygen, okay, high partial pressure of oxygen, it goes to the right. Low partial pressure of oxygen, it will actually go to the left. So let me go to the left first. Okay. So this is a chemical equation, which you know that a chemical equation can be moving to the right or to the left. It actually depends on both the concentration of your substrate, the substrate, versus the concentration of the product, right? Okay. So in this case here, we are looking at loading and unloading of hemoglobin, because that is the main function of hemoglobin. Hemoglobin in our red blood cell function is mainly to load oxygen in the lung and unload oxygen in the tissue. Okay, so therefore, whether hemoglobin will load or unload oxygen, it all depends on the surrounding partial pressure of oxygen. So based on this equation, you can see that in the lungs, right? In the lungs is when there is high partial pressure of oxygen. So this one happens in the lung. You cannot see lungs. L-U-N-G-S, lungs. Okay, so it happens in the lungs. And a low partial pressure happens in the tissue, right? In the tissue. Okay, so this graph is actually representing this equation here. So we start with the lung first. Okay, so we start with the lung first. In the lung is where ventilation will happen, where we keep on taking in fresh air. So they've got high partial pressure of oxygen in the lungs. So due to high partial pressure of oxygen, this chemical reaction will tend to move forward. So when it tends to move forward, it means to say that hemoglobin will actually take up oxygen. So therefore, it is said that when there's high partial pressure of oxygen, hemoglobin will tend to pick up oxygen so that the reaction will move forward. So therefore, we describe hemoglobin as having high, I use red, having high affinity for oxygen. So therefore, when there's high partial pressure of oxygen, hemoglobin will have high affinity for oxygen. So that's why oxygen can bind with hemoglobin to become oxyhemoglobin. Okay, because the substrate concentration is high, partial pressure of oxygen is high. So hemoglobin will bind with oxygen and becomes oxyhemoglobin. So therefore, hemoglobin is considered as having high affinity for oxygen. Okay, so therefore, because of that, oxygen is being loaded. So the loading will happen at high partial pressure of oxygen, which I've got no space to write. Loading, okay, right here, okay? So loading will happen. So this is where exactly we want our hemoglobin to load oxygen in the lungs. And then after that, from the pulmonary artery, which goes to the lung, and from the pulmonary vein, it goes back to the heart, and then it goes to systemic circulation to supply all our organs and tissues with that oxygen. So therefore, now the blood will be traveling from the lung, okay, inside the blood vessels, all the way until it goes to the tissue. So you can see that the oxygen, partial pressure of oxygen from the lung, as the blood is being transported away from the lung, it will start to, the partial pressure of oxygen will start to drop. And of course, it is lowest in the tissue because tissue is made up of cells that will keep on takes up the oxygen, okay, for in respiration, aerobic respiration. So you take up a lot of oxygen for aerobic respiration so that it can produce ATP, so that it can carry out work. So all sorts of work, you name it, that you have learned before. Okay, so that's why the oxygen, partial pressure oxygen in the tissue is low, because the cells keep on taking the oxygen and use it. So the lung is where your father bank in the money for you, okay? And the tissue is where you use the money. So therefore, when the father bank in the money, you got a lot of money. Then you take out and use, become less. All right, so it's like this. Now, let's talk about in the tissue what happened using this equation. So in the tissue, tissue has got, what did I remove? Okay. Tissue has got lower partial pressure of oxygen. So because it's got low partial pressure of oxygen, okay, then you can see that this partial pressure oxygen is lower already. So this reaction will tend to move backwards because of low partial pressure of oxygen or low concentration of substrates, okay? Therefore, this reaction will tend to move to the left. So when it moves to the left, what happened is like this. The hemoglobin will then has got lower affinity for oxygen, and that's why it will start to unload oxygen because it's got lower affinity for oxygen already. So it will unload oxygen. So when it unloads oxygen, so oxygen will be released and made available to our respiring cells. And then hemoglobin will be free again. Okay? So these are the keywords that you need to use to describe the process. If you use this equation, it'll be easier to actually understand and to describe crystal clearly. All right. So now we look at exactly where is it in this graph. We can say that, okay, from at this point onwards, this range is considered as the lung. This range is considered as a tissue. So there's this term called unloading partial pressure of oxygen. Unloading partial pressure means it is the partial pressure of oxygen when our hemoglobin will unload oxygen to our tissue. So unloading partial pressure oxygen is measured at 50% saturation. So therefore, you check from y-axis, when our hemoglobin is 50% saturated with oxygen, where is it located? So it's here, right? And then we interpolate to check your partial pressure oxygen. So partial pressure oxygen here denotes the unloading partial pressure oxygen. So that means from this point onwards, it's considered as the tissue. So therefore, you see here, from this point onwards, all the way down here. So from this point onwards, it is the tissue already. Okay, so this unloading partial pressure of oxygen. So of course, we also want to know where is it the partial pressure that can be found in the lungs. Then this is called the loading partial pressure. Okay, loading partial pressure will be found in the lungs. So how to find out loading partial pressure? You have to look at 95% saturation of hemoglobin. So when the hemoglobin is 95% saturated, that is the loading partial pressure. So we have to find it out from this graph. So at the y-axis, which is the look for 95% saturation of hemoglobin, and then interpolate to the curve on the graph, and then go down to the x-axis and look for the partial pressure of oxygen value. So this is 10 kilopascal. So which means to say that from 10 kilopascal onwards, my hemoglobin will start to load oxygen. That is when it is in the lung. It denotes it is in the lung, so it will have to actually load oxygen because this is the function of hemoglobin. It must load oxygen in the lung, it must unload oxygen in tissue. So therefore, it will load oxygen from 10 kilopascal onwards. Okay, so which means to say that, you see, I'm going to add another line. I'm going to add another line here. All right. Now, do you see this straight line that I've drawn on the same graph? Now, this straight line that I draw on the same graph is for you to compare when the oxygen dissociation curve is S-shaped versus when it is straight line. It is very different. Okay. Now, how different is different? The S-shaped curve is showing us that hemoglobin is very efficient. Okay, hemoglobin is very efficient in loading and unloading oxygen. It's more efficient than the straight line. Okay, than the straight line over here. So how do you check this? Very easy. Every time when you want to compare two curves in this graph, you just have to draw a vertical line downwards. Okay, a vertical line downwards. So from here you check. At 10 kilopascal, at 10 kilopascal, if it's S-shaped curve, my hemoglobin is 95% saturated. Okay, but if it is the straight line curve, okay, my hemoglobin is, how much is this? Okay, so it's roughly here, okay? Roughly here. So probably is 70. So probably this one is 70, roughly I read, okay? So this is, the straight line curve shows that my hemoglobin is 70% saturated. So therefore, which hemoglobin will be more efficient in loading oxygen? Can you type the answer? Which hemoglobin will be more? Yes, the sigmoidal one, right? Chu Jiaying. Yes, the sigmoidal one will be more efficient in carrying oxygen. Hang on. I'm looking for, hang on. You see, so therefore you see that when it's S-shaped curve, right, the hemoglobin is more efficient because at 10 kilopascal already 95% saturated. But for straight line curve, it's only 70%. So not so efficient in loading oxygen when it's supposed to load, isn't it? So this is one way of looking at this. Second way is this. So imagine, for example, now you're in a classroom and the partial pressure of oxygen is, let's say 14. Okay, let's say 14, okay? So both the S-shaped curve and the straight line curve have got the same percentage saturation of hemoglobin with oxygen. So they're the same. But let's say, for example, suddenly there's an electric cut. So no more electricity, no more aircon, and you're still in the room, in the lecture theater, and the door is closed. Is that light or not? Okay, never mind. Suddenly there's no aircon, there's still light. Okay, there's no aircon, the door is closed. So, and then imagine the whole lecture theater got 100 students and all of them are taking in oxygen and releasing CO2. So the oxygen, partial pressure oxygen in the lecture theatre will start to drop, okay? Because so many people take the oxygen and didn't replace back already, right? No ventilation. So the oxygen, partial pressure oxygen will drop. So at that time, you see what happened to our hemoglobin on the S-shaped curve. It is still able to load a lot of oxygen, right? So even though the oxygen level dropped from 14, say for example, to 10, so from probably 98% saturated, it can still load 95% of the oxygen. Very efficient, right? It doesn't really affect much the volume of oxygen that it can load. It can still load a lot. But if you see the straight line, straight away it plunges deep down already. So therefore, instead of 98% saturated, now only 70%. So not efficient at all. All right. So can you see that our hemoglobin is so efficient? It's S-shaped curve. Again, what designed this thing that's so efficient? Amazing, isn't it? Okay. So that is in the lung. What about in the tissue? Now the function, I find that this curve is very difficult to explain. If a curve which is even in the horizontal and go up and even in the horizontal here, then it's easier for me to explain, because when the curve is like this, then I can explain this part more easily. So probably I'll use this one here that I have drawn to illustrate better. Okay, because I find this one's very difficult to illustrate clearly. So now this one is okay already, right? The S. This one is more like, not so S, this one is more S, okay? So this is to show you, let's say, for example, 50% saturation is here. So let's say this is 100, let's say this is 100%. So this is 50% saturated, okay? So 50% saturated is, wow, so high, so high. Okay, never mind. I don't want to use this bit too high. Okay, let me do slightly lower a bit. So let's say, for example, this is 50% saturated here. Never mind, can. So let's say this is 50% saturated. So you can see that this is from tissue onwards, the hemoglobin needs to unload oxygen. So if you compare the S, not so nice. No, cannot. Let me adjust again. Lower better. So let's say, for example, this is 50% saturated. Okay. 50, am I right? Yes. Am I right? No. Okay. No. Never mind. Just use a line to indicate 50%. Is this? Okay. Let's say, for example, don't 50%. Let's say this is 3 kilopascal. Partial pressure oxygen is now at 3 kilopascal in the tissue. Okay. Now at 3 kilopascal in the tissue, you see the red curve and the black color line. The red curve have got lower saturation of hemoglobin. Lower saturation of hemoglobin with oxygen. Black line has got higher saturation of hemoglobin with oxygen. So which one shows that hemoglobin is more efficient in unloading? Can you type the answer? Line curve, the line or the curve? Line curve means what? Line curve means the straight line or the curve? Line means the straight line, is it? Which one is more efficient is in unloading oxygen? Means if they unload a lot of oxygen, they'll be less saturated. When they load a lot of oxygen, they'll be less saturated. So which one is less saturated at 3 kilopascal? Red color line or black color line? Hello, please type your answer. Red color line, right? Yeah, correct. Red color line. Andrea, you get two points. No, 1, because it's after. Okay. All right, so it's 1 because here the red color line you see is actually lower in saturated hemoglobin. that color line is higher in percentage of saturated hemoglobin. So that means in the red color line, the hemoglobin has unloaded a lot of oxygen. That's why there's less saturated hemoglobin. Okay, so therefore, you can see that the hemoglobin in the S-shaped curve is more efficient in unloading at lower partial pressure oxygen than the straight line. So this is to show you that our hemoglobin is very efficient in loading as well as unloading oxygen in the right location in our body. Okay. Okay. So with that, this is actually in words form that explain what I've mentioned just now. So let's go through it to recap. In the lungs, the partial pressure oxygen is high. So oxygen can easily bind to the hemoglobin in the pulmonary capillaries. Therefore, hemoglobin has high affinity for oxygen. So a lot of oxygen will be loaded from the lungs into the hemoglobin. Therefore, there's high level of oxyhemoglobin. So the advantage of S-shaped curve is the affinity of hemoglobin for oxygen is higher than that for straight line curve for a wide range of partial pressure oxygen in the lung. So starting from the partial pressure oxygen when it's 95%, hemoglobin, they are saturated onwards. So from 10 all the way to 14 kilopascal. Okay. So oxygen can be loaded even though partial pressure oxygen drops slightly within this range due to overcrowding, stuffy room, partially obstructed airways. Okay, it will still be able to load quite a lot of oxygen. Now, in the tissue, partial pressure oxygen is low because the cells of the tissue are metabolically very active, so they will take up oxygen for aerobic respiration. So at low partial pressure oxygen, it's difficult for oxygen to bind to hemoglobin at our systemic capillaries. So hemoglobin has low affinity for oxygen here. So there will be a lot of oxygen being unloaded from hemoglobin so that oxygen will be available to the tissue. So the advantage of S-shaped curve here is that a very small drop of partial pressure oxygen can result in the release of large quantity of oxygen. Okay, so you can see here from, let's say that's my 50% here, right? Okay, so from 50% onwards, where is my? So from 50% onwards is the unloading partial pressure here, say for example. So you can see that as the partial pressure drops, you can see the curve also drop very steeply. As the partial pressure of oxygen drop, the red color line, the curve drops very steeply. That means unloading occurs very quickly as the partial pressure drops. So large quantity of oxygen can be unloaded. Okay, a small drop in the partial pressure in the tissue leads to a large release of oxygen. Okay, so that makes the hemoglobin very efficient compared to straight line. You see, a small drop in the partial pressure, yes, there's unloading, but it's not as fast as the red color line. So it's not as efficient as the red color line in unloading. So that's why the red color line is actually more efficient. The S-shaped curve is more efficient than the straight line. Okay, because as the partial pressure oxygen drop, affinity of hemoglobin for oxygen is very low. Okay, for this range of partial pressure in the tissue. Starting from partial pressure of oxygen, when 50% of hemoglobin is saturated. So this point here, probably about 3.6. Okay, maybe 3.6. Yeah, okay. So therefore, oxygen can be unloaded to the tissue even though the partial pressure oxygen in the tissue is slightly high due to low metabolic rate. That means when you are fully resting, sleeping, therefore metabolic rate is slow, usage of oxygen is slow. That means the partial pressure in the tissue is slightly higher here, but your hemoglobin can still unload oxygen. Of course, when you exercise, the partial pressure will be around this area here, right? You use up a lot of oxygen. So oxygen, partial pressure oxygen in the tissue when you exercise will be maybe around here. When you sleep, maybe it'll be around here. Whether it's exercising or resting, your hemoglobin can still unload oxygen because this is what they're supposed to do in the tissue. Okay, so that actually explains to you the efficiency of this S-shaped curve and the location where oxygen will be loaded to the hemoglobin and where oxygen will be unloaded in hemoglobin. Okay, so what about when the oxygen is being transported in the blood vessels. So in the blood vessels, there are a lot of endothelial cells on the wall of facing the lumen, okay, on the wall of capillary facing the lumen, and also white blood cells. So they will use oxygen for respiration. Okay? Do red blood cells carry out aerobic respiration? Anyone wants to answer? No, very good. Rebecca, can you explain, Rebecca, why red blood cell does not carry out aerobic respiration?
It doesn't have ATP.
Again?
It doesn't have ATP.
Enough ATP for 3 months?
It doesn't have ATP.
4 months. 4 months, yeah. It doesn't need ATP, is it?
Yeah.
Yeah, can be correct. It doesn't need ATP. And also, does it have mitochondria?
No.
No, it does not have mitochondria. So how can it carry out aerobic respiration? Okay, so correct. So it does not have mitochondria, so it cannot carry out respiration, so it doesn't need oxygen anyway. So it will not take the oxygen. Okay, so therefore, it's just like a bag carrying hemoglobin and being flowed together with the blood. Whenever there's high level of oxygen, then the oxygen will automatically load to hemoglobin. When it's low level, oxygen automatically unloaded, right? Like the automatic, nowadays got this automatic, like auto-driven buses. Auto-driven buses in some country, right? Okay, not our country yet. Okay, so they will do their function. Okay. So only endothelial cells and white blood cells will be using the oxygen in the blood vessels for respiration. So therefore, when oxygen in the blood vessels is used already, the partial pressure of oxygen will drop. Right, the partial pressure of oxygen in the blood vessel will drop, leading to oxygen being unloaded from oxyhemoglobin. So it will also be unloaded when it's being transported for those cells around it. Okay. Okay. So recap. What does S-shaped oxygen dissociation curve shows? It shows that hemoglobin can rapidly load oxygen in the lungs in a range of partial pressure oxygen and unload oxygen in the tissue also in a range of partial pressure of oxygen in the tissue. And this is all based on the partial pressure in these two locations. Whether loading or unloading will occur depends on partial pressure oxygen. Okay. So now I want to explain to you why that curve is an S-shaped curve and not a straight line. It has to do with this property of the hemoglobin, which we call it allosteric property. Okay, allosteric property of the hemoglobin. Now remember, when you learn the word allosteric, can you all tell me what is the meaning of allosteric? Anyone? If not, I'll call your name. Okay. Sufyan?
Yeah, forgot.
Forgot. Okay. Who wants to try? What is allosteric? Try. No harm trying. Other shape, different side, chain shape. Yeah, correct. Chu Jiaying, correct. Wow, Chu Jiaying collecting a lot of points today. Very good. Okay, please continue collecting. Chu Jiaying, correct. So when you study biology, please pay attention when you first study the word so you remember. Aloe means other. Steric means state. State means other, state means other object. Object means other shape or form. Okay, other shape or form. So usually globular proteins with the binding sites will have this kind of property. the globular proteins, you name it, wherever you can find them in our body, whether it's inside the cell or outside the cell. If it's a globular protein with the binding site, then it will have got this allosteric property. Allosteric means taking other kind of state, means other kind of shape. Okay, so allosteric property. Now, Losisian said different site. Now, it's different site only when you say it is an allosteric site. It's a different site, which is found inside the enzyme for a non-competitive inhibitor to bind. But here, there's no word site. So when there's no word site, it's not the site. So every single word in biology will give you the meaning of the overall meaning. So one word missing or one different word is a different meaning. Okay. So allosteric property means that this protein has got this property whereby when something binds to this protein, okay, let's say this is a protein here. Okay, it's got a binding site. So when something binds to the protein will change shape because it has to hold that structure. So let's say, for example, originally it is this shape, okay? And then something sits into it, almost similar shape. So the induced fit. So now the protein change shape to hold these molecules that it's supposed to be holding. So it has changed shape already. So this is called allosteric property of the globular protein. Okay, so hemoglobin has this property also because hemoglobin is made of 4 globular protein and each of them got a binding site for oxygen. So therefore, it will have this property. So I illustrate this using this shape here, all right? The square and the circle. So what does it show here is when all the 4 subunits are empty, there's no oxygen, I use the 4 square to represent. So when it's empty and no oxygen, the shape of the hemoglobin is such a way that the opening to the pocket is very narrow. The opening, remember I showed you before, this is the alpha chain or beta chain, and right in the center is where your heme group is seated. So we imagine that when all the 4 subunits are empty, this opening here for oxygen to enter is very small. So it's very difficult for the first oxygen to enter. Okay, so even if you see, when you increase the partial pressure oxygen, that means you got more oxygen molecule around it already. Okay, you got more oxygen molecule around it. Only it will increase the chances of the oxygen entering, but very slim. See, if the oxygen are very few, so these 2 are even at the back of this, cannot enter, right? And then you got some in front and some happen to, you add more, then chances of entering is higher. So you can see that as you increase partial pressure of oxygen, the curve will go upwards, meaning to say that oxygen will start to enter the, will start to be loaded, will start to sit into this pocket. So as you increase the partial pressure oxygen, then there'll be higher chances for the oxygen to sit into the pocket, but very difficult because the opening into the pocket is very small. So it's represented by the square when the opening is very small. So after the first oxygen successfully enter through the narrow opening and sit into the pocket already, then of course, this subunit have to hold the oxygen, so it will change shape. So when it changes shape, it's represented by round, okay? And then when it changes shape, its neighbor, which is also attached to it by the 4 different types of bonds, all right? The hydrophobic, from the strongest one, the covalent disulfide bridges, okay? Ionic, hydrogen and hydrophobic interactions, right? So when this one change, when this subunit changes shape, it will cause these 2 neighboring subunit that's attached to it to also change shape, okay? Because it's like two are holding each other together, right? So how to show? When this one change shape, this one also change shape a bit. When this one change shape, it's very, very strange. When this one changes shape, this one, you see, when it's attached, right? When I open my hand, okay? When I close my hand, this one also will be pulled up. So you open already, right? I close my hand, this one pull up. Okay, originally is like this. Originally is like this. Okay, I hold the oxygen and when I close my hand, this one have to open. So both sides will open. So when both sides open, then it's easier. So I can represent like, okay, open, now open already. Okay, open wide. So it's easier for oxygen to enter the pocket. Okay, enter the pocket is easier. Okay, so out of 4 subunits, now there will be 2 subunits that's very easy for oxygen to enter. So out of 4 subunits, 2 are available for oxygen to enter now. Okay, so now your second oxygen molecules enter here. So when it enters here, it changes shape and it causes that neighbor, this neighbor to change shape. So this neighbor now becomes a round. So now in this hemoglobin, out of 4 subunits, there will be 2 subunits available and easy for oxygen to bind. Originally here, you got 4 subunits, but it's almost like 0 out of 4 that's open for oxygen to bind. Can you see that? 0 out of 4 that is open, for oxygen to bind. So very difficult to enter. So therefore the curve is represented by, at the beginning, when oxygen level is very low, or there are no oxygen that are found in the subunit, and therefore the subunits are actually closed. Opening is very narrow, very difficult for oxygen to enter. So when it's very difficult for oxygen to enter, you increase concentration, very little will load oxygen. So very little oxygen is loaded, very difficult to enter. Then in the second case, after the first oxygen is loaded, now the chances of entering is 2 out of 4, because 2 are open already out of 4. This one is already occupied, cannot, saturated. This one is closed. So out of 4 subunits, 2 are open. So 2 out of 4 is of course easier than 0 out of 4. So therefore, when you increase partial pressure oxygen, then there'll be more oxygen to be loaded than before. So it's faster. It goes up faster for the second one. Okay, so this is the first one, and this is the second oxygen. Okay, then the third one also same thing. The third one to enter is easy because there are also 2 subunits open out of 4. The other 2 are saturated. So again, as you increase oxygen, partial pressure oxygen, oxygen will also be loaded very easily. So almost similar gradient, because 2 out of 4 chance. And for the last one, last oxygen to enter is actually easy because this is open. It's easy for the oxygen to enter. But the chances of entering for this hemoglobin is actually 1 out of 4. Okay, for oxygen to enter, this one is easy because it's open, but out of 4 subunits, only 1 is open. So the chances of entering is 1 out of 4. So 1 out of 4 will be slower than these 2, but it's faster than this one. So therefore, it will curve like this. Okay, and then level off. So that explains why the oxygen dissociation curve is an S-shaped curve. It is due to the allosteric property of the hemoglobin. Okay, so first oxygen to enter, very hard because it's 0 out of 4. Second, after the first oxygen enter, second enter is easy. 2 out of 4 is 1 out of 2. Third oxygen to enter is also 2 out of 4 is 1 out of 2. Fourth oxygen to enter is hard, 1 out of 4. So this is very hard and this is hard. Okay. Okay, now I want to show you 2 oxygen dissociation curves at the same time. On the left is the oxygen dissociation curve when there's low partial pressure of CO2. On the right, when it is high partial pressure of CO2. Okay. Now, low partial pressure of CO2 occurs in the lung, isn't it? Because in the lung, that's where CO2 is being removed from our bloodstream. So the blue color curve actually represents in the lung. Okay. And in the right side, the right color curve is when there's high partial pressure oxygen. That means to say that the right color curve is actually indicating it is in the tissue. So you can see that the oxygen dissociation curve is not only depending on the partial pressure oxygen. Okay, this curve does not only depend on partial pressure oxygen, it also depends on the partial pressure of CO2. When it's low, the curve will shift to the left. When the partial pressure of CO2 is high, the curve will shift to the right so that the hemoglobin can load and unload oxygen efficiently. So how are you going to explain this? we'll always, when you have got two curves on the graph, you must draw a vertical line to explain it, okay? So I've drawn a vertical line here. In the same partial pressure of oxygen, you draw the vertical line and you check. For this curve on top, there is a higher saturation of hemoglobin with oxygen compared with this curve at the bottom. So that means to say that if it's the upper curve, if the upper blue color line curve, its ability to load oxygen is better than the lower curve, correct? And the lower curve, its ability to unload oxygen is better than the upper curve. So therefore, it can show you that the S-shaped curve will also shift according to the condition in the body so that the hemoglobin can carry out its function more efficiently. It must load oxygen wherever it needs to load. It must unload oxygen wherever it needs to unload. So you see, so whenever there's high partial pressure oxygen in the tissue, the oxygen dissociation curve will shift to the right side to further cause more oxygen to be unloaded from the hemoglobin. Okay. So this is to ensure that hemoglobin unload oxygen, more oxygen to be made available for the cell to carry out aerobic respiration to produce ATP. Okay. So this is showing you oxygen dissociation curve shifting to the left. When it shifts to the left means that the hemoglobin likes the oxygen, so it loads the oxygen. Okay. Check again, just now. You see from here, if the curve shifts to the left, right? If the curve shifts to the left in the same partial pressure, hemoglobin load more oxygen, right? So to the left means, how do you remember? L, left is L. L means like. Like means hemoglobin will take up oxygen. Okay. Shift to the left means like. So hemoglobin load oxygen. Shift to the right. Okay. Right means R. R means reject. Reject means hemoglobin reject oxygen, so it will unload oxygen. All right. Okay. So when oxygen dissociation curve shift to the right, it shows that our hemoglobin will have got reduced affinity for oxygen because at this region there's a high partial pressure of CO2. So the tissues are highly metabolically active. So oxygen needs to be released to make available for them to carry aerobic respiration, to produce ATP, to carry out work. So when there's high level of oxygen, of course, the pH will also be low because CO2 dissolves in water to become carbonic acid. Okay, so that's why the pH will also be low. So which means to say, in another word, instead of giving you CO2, high level of CO2, I can give you at low pH. What happened to my oxygen dissociation curve? Then you must say that it will shift to the right side. For what purpose? Because this low pH is due to CO2 that dissolve in water to form carbonic acid, H2, CO3, right? Carbonic acid. Carbonic acid. It's the same, isn't it? So instead of giving you CO2, I can give you pH, change in pH. Okay, same thing here. Low CO2, so pH is higher. So I can give you higher pH. A region of the body when pH is higher, what happened to my oxygen dissociation curve? Okay. Now, so therefore, I obtained this from the textbook to show that when you've got two S-shaped curve, this one is when there's when on the left side is when the hemoglobin like oxygen, so loading will occur. So left side belongs to where there's in the lung here. So loading will occur over here in the lung. Okay. Right side means reject. Right side means when hemoglobin rejects oxygen. So unloading must occur. So when you say unloading actually occurs in this curve on the right side over this region here. So when I got these two curves, loading occurs on the upper curve on the right side, unloading occurs on the lower curve on the left side, because the curve will shift to the left and right according to the partial pressure of CO2 also. So therefore, where exactly is in the lung? This part here is in the lung. Where exactly is in the tissue? This part here is in the tissue for this curve. Okay? So this is something new that I find in the textbook. Right? Okay, now let's look at other conditions. So other than partial pressure of oxygen and also partial pressure of CO2 and pH, what else can cause the oxygen dissociation curve to shift to the right and left? See, shift to the right and left, because you can be asked to you can be asked to sketch the curve. They give you a curve already, they ask you to sketch. So what happened? Where will be the curve be now when the temperature increases or when the temperature decreases? Okay, temperature increases is because there are a lot of heat being released, right? When you got a lot of heat, being released, is because you got a lot of respiration, because aerobic respiration produces energy in the form of heat plus ATP. So that means to say that you will need a lot of oxygen. So therefore, to the right is when hemoglobin needs to unload the oxygen. So to the right means reject. Okay, this is for you to remember only, but you cannot write the word reject. To the right means reject. Reject means you will unload. So therefore, it will unload oxygen to make it available to the tissue. Okay, so to the left is opposite lah. All right. Okay, so that's all for today. So I have actually explained to you the meaning of oxygen dissociation curve. And I've also explained to you that the oxygen dissociation curve can shift to the right and left depending on the condition of the body to make oxygen available to the tissue or to load oxygen. Okay, so that's all for today.